Keith Warren here, and on this video we'll tell you about the North American Deer Registry and how important it is to not only maintaining value in your animals, but how advancements with DNA testing are now influencing the deer industry in a very positive way.
North American Deer Registry was started in 2005, and currently we have over 250,000 animals registered within the registry. NADR is a non-profit, non-political organization, and it serves the industry as a whole for all breeders and all producers of cervidae throughout the country. The North American Deer Registry is designed to validate the parentage of individual deer through a DNA analysis process. The DNA is provided to the registry by the breeder and then run through a process that gives us specific information about the genetics of individual animals and compares it to other animals. The reason this is important is because it gives credibility to our industry and it validates the parentage of the animal.
Since 2005, when NADR started, a lot has changed not only in the deer industry, but also how NADR is now using the latest technology to determine parentage. This new technology is called SNP technology, and on this video, you'll be hearing a lot about this new SNP technology and why it is so important for everyone who's breeding deer that have not been converted over to SNP technology should send in samples as soon as possible to have this done. The previous way that NADR determined parentage was called STRs, and the new SNP technology is by far a more accurate way to determine parentage. You'll also see how to submit samples for these older animals that you may be currently breeding that need to be updated to SNP's technology. Something else I'd like to point out, the North American Deer Registry is often referred to in this video as NADR or NADR.
I'm Rob Beckham, and I'm a whitetail conservationist. I've been breeding whitetail deer for about a dozen years. I've been on the NADR board for several years, and we have a herd of about 200 whitetails in Callahan County. I started breeding whitetail about 12 years ago. Nobody knew me from Adam. There were a lot of big breeders out there, and I started using their genetics. And I wanted people to be certain that what they were getting is what I was saying they were getting. And so DNA allows you to do that. I was AIing with some of the best sires out there, and I wanted people to know beyond a shadow of a doubt that they were getting what I was telling them that they were getting. The way you do that is NADR and genetic testing. As I said, I've been on the board several years, and we've been trying to get better and better about how we provide you with the parentage of the animals. And we looked at other industries that have already gone through this process. Most industries do start with STRs, but they move to SNPs because it's more valuable information and has more markers. We can give you a much more fuller picture of what you're getting. When we started, STRs were fine. At the end, we were using 19 markers. But as the industry gets bigger and bigger, more animals following the same genetics, i.e. sires that people are breeding to over and again, means it makes it harder. So if you can look at STRs with 19 markers and SNPs with over 400 markers, it's kind of equivalent of a grainy black and white picture versus a high definition 4K picture.
In order to do this SNPs research, NADR relies on a genetic laboratory located in Lincoln, Nebraska, named GeneSeq.
GeneSeq is a company that focuses on molecular testing, and the data that is utilized based on these molecular tests are used to create informed decisions to make breeding selection. They would be making breeding selection decisions in cattle and chickens and sheep, dogs. Many different types of organisms would utilize molecular genetics in order to make the best breeding decisions. The number one goal that GeneSeq has for the North American Deer Registry is to utilize DNA information to verify parentage. Secondly, we can utilize molecular genetics to look at disease resistance. So diseases such as chronic wasting disease. We can evaluate the underlying genetic characteristics of those animals to determine if they may be more susceptible to those diseases. Third most important thing that we are able to do here is look at the entire genetic potential of an animal to ensure that they're passing on the right genetic characteristics on dozens of different traits to the offspring and make those types of informed breeding decisions. There are two major things that we're utilizing in the lab to reduce turnaround time to the North American Deer Registry customer base. That would basically be conversion to TSU tags that better enable automation for DNA extraction and downstream sample handling, and also converting to a SNP-based technology where we can use robotics to enable quicker data collection, but also data scoring is much more accurate than the traditional microsatellite or STR approach. STR is now really considered to be dead-end technology. Every animal breeding association that we work with has been transitioning to SNPs for the last 10 years or so, including the North American Deer Registry. SNP technology is much more accurate to interrogate pedigree accuracies. The basic reason why is because there are millions of SNPs that span the entire genome. With microsatellites, there's only a few thousand that have been identified for every individual. So we have the choice to include as many data points as we need to accurately integrate the structure of the pedigree. I like to look at SNPs as kind of the gateway drug into more sophisticated genetic testing. Not only does it enable us to test parentage with more accuracy and inbred lines, but it enables us to evaluate disease resistance such as CWD, EHD, and other things that may become more complex in the future, including certain traits like feed efficiency, the size of racks, body conformation, a lot of complex traits where we can evaluate the entire genome of an animal rather than something very specific. Traditionally, the primary sample type that would arrive in the lab from the North American Deer Registry has been a hair sample. The hair sample is proven to be cumbersome in the lab. It's also more prone to contamination, not only in the field, but in lab processing as well. Some of the challenges include small follicle size and static electricity. Another type of tissue sample or sample that we get for North American Deer Registry samples is what's called a TSU tube. And the TSU tube is tissue sampling unit. It's available from Allflex, or it can also be purchased directly from GeneSeek. The kit is easy to use. It comes in a kit of 10 vials. Each of the individual tubes are barcoded. It comes with a handy applicator to apply the tags and to collect this tissue sample, and it's easy to match the animal ID back to the sample ID. Once the producer gets the sample, they simply put it in the mail and mail it to the North American Deer Registry.
Now we'll show you how tissue samples are collected from fawns at a Texas deer breeding facility.
Oh, she got a big old belly. So the first thing we're gonna do is set her in here and set it in here and see what it is. Hey girl, you're all right. Make sure that it is what we think. It is a little girl.
So we get our next doe tag available, which is 9002, and we get our next available unique number. And from there, we will record this information on our spreadsheet for all of our fawns. Her unique number is AR5G. She was born on May 28th of 19. Her sire is Blackjack. Her mother is Pink 2017. And the dam sire is Express. And they were born in pen 12.
So the next thing we're gonna do is get our tissue sample. So here's our applicator. Here's our box of tubes. We open it up, we pull the next tube out, put the tube in the applicator. Just pull this little piece out. Save that for later. So we take our tissue samples out of the same ear that we tag in, this right ear here. So this piece pushes the sample into the tube here. So after this, we take the tube here, put it back on this little red piece here so you can secure it to your box. We write the tag number, unique number, and just punch it right here, and it'll be ready to send off once you get all these filled in.
Now after that's done, we are ready to put the tag in. So we'll load the tagging gun with the back, the front, and then from there we disinfect the back with alcohol. And we'll come over here and we'll put the tag in the same hole that we collected the tissue sample out of. Now that all that's over with, he'll give her some fawn paste and we'll release her back to her mother. Find her. Little girl.
Like I mentioned earlier in this video, it's important that if you're breeding with animals that have not been converted to SNPs technology, that you get these breeding animals converted from STRs to SNPs. And here's how to do that. So converting your deer over to SNPs can be intimidating, and it is a timely process, but I'm in the process because I know that in order to stay up and stay in the game in the deer breeding industry, we all need to be in the SNPs program. So what we thought we'd do is show you how I'm going about doing this. I have 233 deer I'm fixing to do. Okay, and so what I've done, I've got my deer separated as far as age groups here, the oldest and then the next one all the way to the youngest right here. These are all the deer in my breeding program right here. What I've done is I've pulled a sample off of every single deer that I have, and on the sample you'll notice it has a unique number, it has a year of birth on it, and also on the back side I've got a NADR number. In other words, these deer are currently in the registry. Now, the deer that are not in the registry, those are going to be done last. But what I'm going to do, I'm going to bag these up and I'm going to sort them out and bag them up into age groups. And I'm going to send them all in with nice notes. And then I'm going to also send in a list of the bucks that I've used to breed over the years. Okay, and I've got their NADR numbers on here. Get it over to the folks over there at NADR in Edmond, Oklahoma. And it's a matter of waiting to see what I come back with. So to make things easier on the ladies when it gets up there to the main office, I'm just going to take and sort these out, put them in little plastic bags, and then write the year they were born on the outside of the bag. Just makes things a little bit easier and more organized for the ladies when it gets up to the main office. And make sure you include the instructions with it. Most animals that have been sent in to NADR for testing since 2017 are already done using SNPs. If you have animals that you aren't sure whether or not they've been SNPed or converted to SNPs, log on to the NADR website, and on your inventory page, look to the far right side. And if the animal has been SNPed, it will say complete. If it says none, and if you're still breeding that animal or plan to breed that animal in the future, you should get samples and send them in to NADR so that it can get SNPed.
My name is Leah Segrist. I work here at NADR. My job here at NADR is to process the samples. So once I receive the samples, I place them into the computer. I find the farm name and make sure that we have the correct farm, put the animals into the database. We generate a number at that point and gives you your NADR number. Once we do that, we give them barcodes and send them off to the lab.
Now, besides benefiting from increased accuracy with SNPs technology, you'll also benefit as the price per sample will drop. Turnaround time for this process is typically 12 to 14 business days, but that can change depending upon volume of samples being submitted.
Samples are submitted to the North American Deer Registry's office, and they're consolidated into a single batch. Every week, a batch shipment comes to our laboratory in Lincoln, Nebraska. The box is opened, a sample is removed from the box. The sample will then have a barcode that is scanned. That will enable us to determine the actual order number that was created from the North American Deer Registry. From there, we'll take that and scan it for accuracy and make sure that every sample arrived. If a hair sample arrives, we'll remove the hair from the envelope and we'll apply it into a specialized device called a hair sample collector. That hair sample collector enables us to process that sample in the lab. Once the sample has been received, entered into our laboratory information management system, and barcode scanned, it will come to the DNA extraction lab. Once in the DNA extraction lab, the card will come out. The hair follicles will be individually isolated and deposited into a 96-well plate for DNA purification. Once a TSU sample comes into the lab, the first step is to remove the cap. So enabled to do that, we batch a group of 96 samples into a rack. We use a 2D barcode scanner to scan the entire rack of 96 samples, and then a robotic unit will remove the cap so that we can localize the purified DNA. Once the tissue samples and hair follicles have been isolated into 96 well plates, we can begin the process of DNA purification. The first step is to utilize a lysis material that break opens the cell wall. The DNA is then attached to a magnetic bead that we can use to help the purification process for downstream processing. The next step in the process is to create a PCR master mix. PCR is a process that enables us to localize a specific piece of DNA and make billions of copies of that DNA material for the next step analysis. We will then take the DNA material for placement in a specialized instrument. This specialized machine will create billions of pieces of targeted DNA. The amplified DNA material is now spotted on a specialized glass chip. The DNA material that was embedded in the glass chip will be shot with a high-energy nitrogen laser that will enable the DNA to be differentiated by size. Once the data have finished being collected, it will be evaluated for optimal integrity by 2 different data technicians. Results will then be sent to the North American Deer Registry for further evaluation. Samples will be stored indefinitely at -20 degrees Celsius, and tissue samples that can be kept at room temperature storage will be done so in the warehouse.
As you can see, the technology that is required to process these SNP samples is extremely sophisticated and very accurate in comparison to the old way of using STRs. Deer breeders have realized that SNP's technology truly is the best way to validate parentage, and it's this new information that opens up an entire new future for the deer industry, and deer breeders are excited about it.
SNPs are the only way to move the industry forward. There are so many things that we can learn from the DNA of these animals, and SNPs is the cutting edge science on how you find those things out. It's not all about the antlers. You know, we have many different deer species, and people look at many different things about deer. One can be conformation of the animal, the ability to produce milk, the body weight, the overall length of the animal. There are a whole bunch of things we can study.
First and foremost, we want to provide parentage. But we can look at disease resistance. We can breed CWD out of the herd if we can find the markers that are attributed to CWD. The technology is going to allow us, if we choose, to do almost anything we want that other breeding programs are already doing.
SNP's technology has been embraced by mule deer breeders as well, and they're experiencing the same results.
My name's Abraham Garcia, and we work with desert mule deer in Mexico. We're part of Desert Mule Deer Partners, and we've been working with conservation of the desert mule deer and improving genetics and just being able to produce better trophies. So NADR has played a big role in helping us with technology through parentage DNA. The accuracy in SNPs that has come has been a tremendous help to us to be able to do genetic selection and be able to make better decisions on what we're looking at as far as who is who, and to be able to differentiate and produce better trophies and make better decisions in our breeding program.
SNP's technology is not only the best way to determine parentage for deer breeders, it's SNP's technology that'll help take our deer industry into the future using science to help us build better animals for a better future. The North American Deer Registry works hard to offer the best of what the deer industry needs in the genetic registry, and NADR is leading the way for the entire deer breeding industry with this new SNP's technology. In closing, if you're breeding or plan to breed animals that have not been SNP'd, now is the time to get the samples into NADR. By doing so, you'll be up to speed with the latest and greatest technology there is available in regards to genetic testing deer. My name is Keith Warren, and thanks for watching.